Functional human brain tissue is being successfully 3D printed by researchers. Greetings to everyone. In today's video, we will be discussing an exciting breakthrough from the University of Wisconsin-Madison. Scientists there have successfully created 3D printed brain tissue that exhibits the same growth and functionality as natural brain tissue. To achieve this, the researchers utilized a specialized bio-ink gel, which served as a soft foundation for positioning brain cells. These cells, derived from induced pluripotent stem cells, were arranged in a manner similar to pencils on a tabletop. Remarkably, the cells were able to communicate, transmit signals, and interact with each other through neurotransmitters. Additionally, by introducing support cells into the printed tissue, the researchers were able to establish proper networks within the brain tissue. This innovative printing technique offers a level of precision that surpasses traditional brain organoids, which are used for studying the brain. Furthermore, it holds immense potential for advancing research on various neurological and neurodevelopmental disorders. Without further delay, let us dive into our discussion on this groundbreaking development. The University of Wisconsin-Madison researchers have successfully created 3D printed brain tissue that exhibits the growth and functionality akin to that of normal brain tissue. This accomplishment holds significant implications for scientists who are engaged in the study of the brain and are actively researching treatments for a diverse array of neurological and neurodevelopmental disorders, including Alzheimer's disease and Parkinson's disease. According to Su-Chun Zhang, a professor of neuroscience and neurology at UW-Madison's Weizmann Center, this model has the potential to be an immensely influential tool in comprehending the communication between brain cells and different regions of the brain in humans. Its impact could extend to revolutionizing our approach to stem cell biology, neuroscience, and understanding the development of various neurological and psychiatric disorders. Zhang and Yuanwei Yan, a scientist working alongside Zhang, have found that the success of previous endeavors to print brain tissue has been constrained by printing techniques. The team responsible for the innovative 3D printing technique recently published a detailed account of their method in the journal Cell Stem Cell. In a departure from the conventional method of 3D printing, which involves stacking layers vertically, the researchers opted for a horizontal approach. They utilized a softer gel known as bio-ink to house brain cells that were derived from induced pluripotent stem cells. This gel differed from previous attempts at 3D printing in terms of its composition. According to Zhang, the tissue retains sufficient structure to maintain its integrity while still being soft enough to facilitate neuronal growth and communication between cells. The cells are arranged in a manner similar to pencils laid side by side on a tabletop. According to Yan, the thinness of our tissue allows for sufficient oxygen and nutrient supply to the neurons from the growth media. The evidence is clear. The cells possess the ability to communicate with one another. Through the act of printing, these cells establish connections not only within each individual layer, but also between layers, creating intricate networks reminiscent of the human brain. The neurons engage in communication, transmitting signals and interacting with one another through neurotransmitters. Additionally, they establish functional networks with the assistance of support cells that were introduced into the printed tissue. According to Zhang, the results of our experiment were truly remarkable. After printing both the cerebral cortex and the striatum, we made an intriguing discovery. Despite the fact that we used cells from different regions of the brain, they still exhibited a remarkable ability to communicate with each other in a unique and precise manner. Brain organoids, which are miniature organs used for studying the brain, lack the precision and control over cell types and arrangement that the printing technique provides. These organoids grow in a less organized and controlled manner. According to Zhang, our laboratory possesses a unique capability, the ability to generate a wide range of neurons at any given moment. Moreover, we have the freedom to assemble these neurons in any manner we desire. By employing this printing technique, we can create tissue with precision, allowing us to closely examine the functionality of the human brain network. With this approach, we can focus on the intricate communication between nerve cells in specific scenarios, as we have the ability to print the exact components we require. Flexibility is inherent in the level of specificity offered by this printed brain tissue. It opens up a multitude of possibilities, such as investigating cell signaling in individuals with Down syndrome, examining the interplay between healthy tissue and neighboring tissue impacted by Alzheimer's, conducting trials for potential new drugs, or observing the growth of the brain. In previous times, our focus was often limited to singular elements, causing us to overlook crucial components. However, our brain functions as a network with cells communicating and collaborating. This is precisely why we aim to replicate brain tissue in this manner, to capture the intricate interconnectedness. Zhang explains that comprehending the brain necessitates studying it as a whole entity. The brain tissue we are developing holds immense potential for investigating various areas of research at the Weizmann Center. It can shed light on the molecular mechanisms involved in brain development, human development, developmental disabilities, neurodegenerative disorders, and much more. Many labs will have the opportunity to utilize the new printing method, as it does not necessitate specialized bioprinting machinery or intricate tissue preservation techniques. The technique can be thoroughly examined using commonplace microscopes, standard imaging methods, and electrodes that are already widely used in the field. The researchers aim to delve into the possibilities of specialization, dedicating their efforts to enhancing their bioink and fine-tuning their equipment in order to enable precise cell orientations in their printed tissue. According to Yan, our current printer is a commercially available model that sits on a benchtop. However, we have the capability to make customized enhancements that will enable us to print specific varieties of brain tissue whenever we need to. Partial support for this study was provided by various organizations, including NIH-NINDS, the National Medical Research Council of Singapore, the Ministry of Education of Singapore, Aligning Science Across Parkinson's, the Bleezer Family Foundation, and the Buster Foundation. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.